Hey everybody, I hope you're doing well this week. If this is your 8th online class of the day, I need you to do me a favor. I need you to pause this video right now and go take a walk. Go out for 15 minutes, do something for you, and then come back to this video with a fresh mind. We know that it's very, very important. And we have a lot to cover today, but it should go pretty quickly. So we're going to start out today, because the action potential is so important, by just quickly reviewing this and taking a walk through it again. So remember, our starting point for the action potential is that some signal comes into some neuron somewhere in the brain, right? We have a signal coming from another neuron, usually in the form of a neurotransmitter. And this signal is going to try and depolarize the neuron that we're looking at until it hits this threshold of excitation. Let me get my pointer out here. The threshold of excitation that will allow the neuron to actually open all of these voltage-gated sodium channels and start to create what we see as the rising point at the action potential. And remember, the numbers here correspond to these numbers up here of what's happening with different sodium and potassium channels. Part of the way up, potassium channel is open, and this allows potassium to leave the cell. It slows down the slope a little bit and becomes very important on the falling phase of this. At some point, the sodium channels become refractory. They hit a point where they are voltage gated, so they have a voltage that causes them to open, but they also have a voltage that causes them to close and become refractory. And so at this point, they're inactivated and they can't be opened anymore. And this is what causes the downward spiral on the roller coaster. And then these voltage gated potassium channels still remain open, and this allows potassium to continue leaving the cell until they hit a point where they close. But they take a little too long to close, and they don't all close at once. So we get this after hyperpolarization, which has a number of important roles, but one of them is controlling how quickly a neuron can fire an action potential while it resets to equilibrium. And that's probably the most important role that it plays, is in the timing of action potentials. And eventually, we are going to get back to this normal threshold that we call the resting membrane potential. So now we have to talk a little bit about synapses, right? Because we've talked about how the action potential works, and then, of course, what happens at the end of the action potential is that a neuron is going to release neurotransmitter. But we have not talked at all about how that happens. So we have these long-range signals, and they are electrical, and they get out to this terminal button, which you should know very well from having listened to the other lectures, is kind of the point of neurotransmitter release. And what happens at this terminal button is that we are going to have a synapse with another neuron. And this is basically a connection between the two. So this junction is very, very small. In fact, even if we take some of the most powerful microscopes that we have in the world, electron microscopes that can zoom in sometimes 16 or 20,000 or even more times to be able to look at very, very small bits of tissue. What we see here is something that looks thinner than a hair. It's as if the neurons are meshed together. And so this synapse, being the junction between the two, is capable of the process of synaptic transmission. And this is basically the notion that a message is being passed between our presynaptic cell, or the one that's sending the message, and the postsynaptic neuron, which is the one that's receiving the message. So when we started out with that idea of the action potential, looking at step 0, we're starting from the perspective of a neuron receiving the message. And today, we're then looking at the message that's sent out, right? We're looking at the point when the action potential gets down to this terminal button, and that we can send some kind of a message. Something that's really important for us to talk about, and it's going to become even more apparent why this is important by the end of the lecture, is that there are many different types of synapse. The reason that these are important is because of the different weights that they carry. So one kind of synapse that we can have is an axodendritic synapse. And so I want to break this down for a second, because it makes it important if the terminology that we use is very, very clear. So the reason why we call this an axodendritic synapse is because if we are looking at the cell that's sending the message, its axon makes a synapse with the dendrite of another neuron. So axo for axon, dendritic for dendrite. So really, we're just looking at these in terms of who is sending and who is receiving the message. And this is by far the most common type of synapse that you will see in the brain. As we talked about in the first lecture on chapter 2, the axons are the primary message for a neuron sending a signal. This is not exclusive, but primary message by which a neuron sends a signal, or the output. And the dendrites are the input. So this is a typical kind of output from one neuron being received by the input of another. We can also have axosomatic synapses, where the axon of one cell targets the soma, or cell body, of another cell. And you should know both of those terms at this point. So an axosomatic synapse for an axon connecting to a soma. And the reason that we care about this synapse is because it's the one that is thought to have the greatest weight in terms of how it influences the processing that the soma does. Third, we can have an axo-axonic synapse, and these are probably one of the more rare types of synapse, where we have to bring in another neuron. And we talked about the idea that action potentials are really difficult to stop once they're started, and that's true. But one of the things another neuron can do is influence what happens at the synapse itself. And so in some cases, we have an axon of a neuron coming in and making a synapse on the axon of another neuron in order to influence the way that it releases neurotransmitter. And so we think about this as being positioned for very specific kind of responses. One of the things that they could be doing is gating the response at this at this synapse. So how do we get neurotransmitters? Where do they come from, and how are they released, and what's the process by which they're released? In terms of the synthesis and storage of neurotransmitters, this is the point where all of the organelles that we talked about become important, because their individual jobs are not quite as important as the role that they play when they all work together. For example, we talked about the rough endoplasmic reticulum, where we have ribosomes that help to make proteins, for example, the proteins that make up neurotransmitters that we need in order to release a chemical signal from the neuron. And then somewhere between what we see as steps 1 and 2 here, we also have the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum makes all kinds of lipid membranes. And we said, well, we need those for, of course, the cell membrane itself. But these are also the outer layer of synaptic vesicles. And the reason for that is going to become very apparent in a few slides here. And then we have the Golgi apparatus, right? The Golgi apparatus is the ultimate in packaging organelles, and it starts to put all of these pieces together, and so they're produced at this step. So when we have all these components, we take smooth and rough endoplasmic reticulum, we put everything in the Golgi apparatus, we combine these proteins into a complex, and this includes taking neurotransmitter and the lipid layers and putting them together into synaptic vesicles for release. And then once these are formed, one of the things that we talked about is axoplasmic transport. And in this case, you should be thinking, hold it in your head, which kind is it? 5, 4, 3, 2, 1. This is anterograde, which goes along with the direction that a neuron signals. So we have the forward direction, or anterograde axoplasmic transport, which is the faster of the two, that carries this out to the axon terminals, or the terminal buttons, whichever terminology you would like to use. And so this accounts for the way that we get new vesicles. And this is pretty important. We produce a lot of new vesicles because they become degraded. Some of them are released and taken back up by glial cells and things like that. And we also have the neurons working to recycle some of these. So what finally happens when this action potential reaches the terminal button? Well, what we have is a multi-step process that is triggered essentially by the electrical activity that's carried down this axon out to the terminal button. And the first thing that happens is when the action potential gets here is that we have specialized ion channels called calcium channels. And if you look back at the table that we talked about last class, and it shows kind of the balance of different ions inside and outside of the cell. One of the things you're going to realize is that calcium is really high outside the cell compared to inside the cell. But we're talking about very little calcium, like 1 to 2 molecules versus a fraction of a molecule inside the cell. So calcium is kept out of the neuron for the most part, almost completely. And when it comes in, it usually has very, very specialized jobs, and there's not much of it. So again, when it gets used, the jobs tend to be very specialized. So in this case, when an action potential reaches the terminal button, it causes these calcium channels to open. And the entry of calcium causes something to happen. So at this terminal button, we might have a lot of synaptic vesicles floating around, and calcium causes them to bind to these fusion pores and starts a process for these fusion pores to open. So what you can see here is something that's very representative of what happens. We have a number of different kinds of proteins. One set of them are called snare proteins, perfectly named because they grab onto the vesicles and help pull them to the membrane. And so these fusion pores with their proteins that grab onto these vesicles then give us a picture of why it was so important that these synaptic vesicles were made up of a lipid bilayer. Because what ends up happening is that the fusion pores pull open and the vesicle, or what used to be the vesicle, essentially becomes a part of the membrane for a period of time, except that we open up this little hole to allow the vesicle to start releasing neurotransmitter. And so the wider this opens, of course, the more the neurotransmitter is forced out. And that means that, remember, the other neuron that we're talking to is located incredibly close to this. So this neurotransmitter is released out in a way that we think is passive, although we're learning more about this kind of thing every day, and will bind kind of at random to the receptors on the other neuron that's receiving the message that would be right where all this text is at the bottom here. So to give you a picture of kind of what that looks like, in the presynaptic cell, we have some kind of a binding of the synaptic vesicle, and we're going to have these fusion pores opening, and of course that has neurotransmitter, and then that neurotransmitter is going to enter into the synaptic cleft, the space between the neurons, which is nowhere near as big as what you see here. I'll try, and if somebody reminds me in the discussion, I'll post a picture. I have a lot of pictures from an electron microscope where you can see some of these components. So if somebody reminds me, I will post a picture, or we might have some coming up in chapter 5 to show you just how small that this synaptic cleft looks like at a 16,000 or 20,000 times magnification. And then what ends up happening is that the neurotransmitter that's released can bind to a receptor on the postsynaptic cell, and that allows ions to enter, and that's what causes the depolarization that we see at the beginning of the action potential. So in many ways, in looking at this, you should now start to think, okay, I've come full circle, because if you go back to the action potential slides and you start at step 0, that starts with something binding, and ions entering the cell to depolarize it. And then that would loop all the way around back to here to the point where the action potential reaches. We have calcium influx and the synaptic vesicle binding, and voila, all of a sudden you have the full idea of a circuit, which is going to go through this input-output, or input processing and output over and over and over again. One other thing that I think is really important, and I'm going to go on a very small rant here, but in talking about the way that synaptic vesicles work, is that there are many different things that a neuron can do with these. So of course, I hinted at the idea that many of these neurotransmitters get recycled. And so this process is pretty simple, and we're going to talk about it a little bit more, where we have neurotransmitter in a vesicle, the vesicle fuses with a membrane, and then there are a couple of different ways that we can take it back up into the cell. And so, of course, we have neurotransmitter that's released, and then we have reuptake through a process called endocytosis, where we either pick some neurotransmitter back up in something that kind of looks like a vesicle, or sometimes there are even reuptake proteins, almost like an ion channel that's on the cell that helps us with reuptake. So if any of you are super nerds in your free time like I am, and you have sat there and said, oh, I wonder what Dr. Barker does in his lab. One of the things that you might have realized is that I spent some time at the NIH working on a new group of neurons that was discovered that release multiple neurotransmitters. And we've known about some of these that do this for a long time, where they have most neurotransmitters are very small molecules, and we've known that there are some neurons that also release vesicles that we call dense core vesicles that have these really complicated proteins called neuropeptides that can also signal. But another thing to point out is that we have these transport proteins, and one of their jobs is something I think the book talks about, which is that they help to recycle neurotransmitter back into synaptic vesicles because we have specific proteins that package neurotransmitter into vesicles. But another really important thing to point out is that these transport proteins are very specific to either a class or a single neurotransmitter. And in many cases, it's a single neurotransmitter. And that means they not only have a job of helping to recycle, but they play a job of kind of sorting, which means that these neurons that we were looking at the NIH that release both glutamate, which if you've thought about anything, this is a neurotransmitter that that activates other neurons, and they also release GABA, which is typically thought to inhibit other neurons. So these neurons we've been looking at are kind of a walking juxtaposition where they should kind of cancel each other out. And of course they don't do that. They have some kind of cool and interesting functions that we're still learning more about. But the transport proteins are important because they help to recycle neurotransmitter, but they also help to segregate and organize, and they play a very active role in what, or at least we think an active role in what certain neurons are doing. On the postsynaptic side, we have a lot of different receptors, and one thing that I want you to realize is that there's kind of a blend of what happens. So we have a lot of different types of receptors. It's one of these things where you could have multiple permutations, because the first thing that we have is what ligand binds to the receptor, right? So ligand is just a fancy term, kind of a catch-all term for neurotransmitters, but also for drugs or those neuropeptides that I talked about, or any number of other things like that. So really any chemical that can activate a receptor would be considered a ligand. And then the other thing is that we talked about this a little bit already, is that these ion channels are very specific. So if you think about just a few permutations, let's say that the glutamate and GABA that I was just talking about, you might have GABA that is the ligand for all of these channels here. But I could have a specific GABA receptor that allows sodium in, or calcium in, or potassium out or in, or chloride, to come in. And then I could have the same thing for glutamate. So already that's 8 different classes of receptors. And this is kind of the way that these things work. They're very specific to both the ion that they allow in, but in most cases they're specific to the ligand or class of ligands that are able to activate and open them. With that in mind, there are 2 main classes of receptors, and I'm going to put these both up because I want you to see them at once. I'm usually the kind of guy that will put something up on a slide and I'll hide the other part so you're focusing on what I'm thinking about. So don't get overwhelmed, because we're going to talk about each of these piece by piece. The first major class of receptors are ionotropic receptors. Okay? And what I want you to think about with these is that they allow ions in, right? Ion for ionotropic. And basically what that means is they look a lot like the receptors I showed you on the last page. A ligand hits them, they open up an ion channel, and ions go directly in. And depending on what ion is going in, or in some cases out, then they directly change whether the cell becomes depolarized or whether the cell becomes hyperpolarized. The second class of receptor we call metabotropic receptors. So the major difference with these, and when you see metabotropic, I want you to start thinking metabolism, right? Which you all know to be kind of a slow chemical process where we have to take food that comes in, if you want to use that as the analogy for the signal, and we have to digest it and we have to process it before we can do anything with it. So when you see metabotropic, I want you to think metabolism. I want you to think long process. This is essentially what these receptors do. So we consider them to be an indirect method of activation. So ionotropic, the second that a receptor has a ligand bind to it, pop, that ion channel goes open. However, the metabotropic receptors will bind, and what they create is this entire long chain of chemical processes. And some of them can work pretty quickly, but they're still not instantaneous. Where and for this reason, that they have to go through this kind of handoff, we call them second messenger systems, because the neurotransmitter binds, and we think of this as being the first messenger. And then what it does is it activates usually a G protein inside the membrane, and this protein basically acts as a second messenger in order to start activating enzymes and things that work within the cell. And the ultimate goal of both of them tends to be the same. They still end up usually opening up an ion channel, but the process is much slower. And an important second piece of this is because the process is much slower, it's also a whole lot longer lasting. So this means that ionotropic receptors are meant to say you either should or you shouldn't fire an action potential right now. But a good way to think about metabotropic receptors is that they're the kind of thing that can change state for a long time, right? If you were to think about hungry versus not hungry, in keeping this idea of metabolism, then those are two very different states in the way that the body feels and that it works and the way that we act and all kinds of things like that. And so if we want to make the cell more depolarized for a very long window of time, maybe a few minutes or maybe even a couple of hours, then we can activate these metabotropic receptors and continue to keep them activated. So for the most part, things are probably on the shorter end of that, but ionotropic receptors are instant. Things bind and then they shut down, and that's the way that they work. And the metabotropic are much longer lasting. So we hinted at this in the action potential lecture, but one of the things that happens once we have these chemical messages and they're received by some sort of a receptor is that there are really two results on the cell that's receiving the message. So the first of these is an excitatory postsynaptic potential. So what this means is that we have the neurotransmitter that binds, and then we have something like sodium enter the cell, and it causes the postsynaptic cell to become excited or depolarized. You can think of both of those in the exact same way. And whether that depolarization is sufficient or enough to fire an action potential, we don't necessarily know, or in the context we're talking about here, we don't necessarily care. Because what happens very locally at the dendrite that's receiving this is we get this excitatory postsynaptic potential. And you could think of it as being, well, potential because it has the potential to cause, in this case, an action potential, but it may not, right? It's just, at this point, it's just a very local depolarization of some dendrite. And we need many of these to come together to reach that threshold of excitation. And the opposite can also be true, where we can have an inhibitory postsynaptic potential. And in this case, we usually have, again, a neurotransmitter is released, but in this case, maybe it binds to a receptor that allows chloride in. And chloride, being a negatively charged ion, comes into the cell and it creates an inhibitory postsynaptic potential or a hyperpolarization. So these postsynaptic potentials, again, probably the best way to think about them is being very local, even though we can measure them at the whole cell level. But for this case, that's what's happening just where this message is received, is that we've inhibited this little segment of dendrite that's receiving the message. So if we go back to this figure that's very similar to the one that we talked about for what happens with diffusion and with electrostatic forces. These are very, very important things. So for the most part, what's going to happen with a given ion for a given receptor is going to depend on which ion channel is open. In the case of sodium, it's a positively charged ion that enters in. And so that means that the inside of the cell, which starts out more negative, becomes more positive. So we get an excitatory postsynaptic potential. If we open up a potassium channel, then what we have in this case is one of the cases where the electrostatic pressure and diffusion work against each other. And the strongest of the two forces tends to be the diffusion force, because potassium is at such a high concentration inside the cell, it really wants to leak out, which means that we have a positive ion leaving the cell. And that means that we're taking away a positive and making the inside of the cell more negative. So this results in an inhibitory postsynaptic potential, or IPSP. If we bind to an ion channel that allows chloride channels to open, and we have some neurotransmitter bind here, then chloride, a negatively charged ion, enters, and this influx will cause hyperpolarization or an inhibitory postsynaptic potential. And then we've already talked about calcium a little bit as being kind of the special case. And so what I'm going to tell you is not to focus too much on calcium here, except to say that as we get through the semester, we're going to see that fusion to open up a calcium channel, there's not much calcium outside of the cell, right? So even if we do open a calcium channel, it's usually not the case that it's going to cause a huge excitatory postsynaptic potential. And what we think of instead is that calcium tends to play very special roles. It does things like help with memory formation by changing the structure at dendritic spines, or it helps to modify the release of neurotransmitter like we just saw at the synapse. So when you think about calcium, you should be thinking about kind of special roles. And for this reason, it's something that you should pay attention to the idea here, but not necessarily something that you should expect to worry about for quizzes and things like that too much. Okay, so how do we end all of these synaptic signals? What's the, where does this all lead to? Well, there are a few different processes at work here. And one of the things that I'm going to say is that your book goes into quite a bit of depth on some of these pieces. And honestly, over the years, what I've decided is that if anything, we need to simplify this just a little bit. So let's talk about the ways that we get rid of neurotransmitter once it's released. The first of these I already talked about a little bit. It's reuptake. So we have the neurotransmitter that's taken back up into the presynaptic cell. And the way that this happens for the most part is that it's removed from the synaptic cleft so that it can be recycled back in this presynaptic neuron. And there are a number of different, like I said, transport molecules, or some little bit of this happens passively when we have these extra pieces of membrane that are taken back in to the cell in a process we sometimes call bulk endocytosis. So that's a little bit of what we know to happen, right? And the whole goal here is to really limit the amount of time that this message is being sent to the postsynaptic cell. So we don't want all this neurotransmitter just sending away forever. So we bring the neurotransmitter back in and actively transport it back into the cell, or sometimes kind of passively or accidentally take some of it back up in as we prepare to recycle vesicles. The other major process is a process called enzymatic deactivation. So in enzymatic deactivation, we have enzymes that actually destroy neurotransmitter. And this happens a lot more for peptides, but it also happens for a few neurotransmitters. In fact, when we talked about the history of physio, we talked about this. The first neurotransmitter discovered, acetylcholine, and it is one that happens to be affected by enzymatic deactivation because there's an enzyme called acetylcholinesterase, say that 3 times fast, that is there to destroy a lot of the acetylcholine. And another thing that I think your book maybe doesn't put as much emphasis on is something that I've made a big point, which is the influence of glial cells. So we've talked about the fact that glial cells, mostly astrocytes, can modulate neurotransmitter clearance because we now know that they take up neurotransmitter and can help with this recycling or reuptake process. So they also play a role here where they can take up neurotransmitters. They're most active at doing this with excitatory neurotransmitters like glutamate that can be toxic if they stay at the synapse for a long time. But this is more of an active process than we ever thought, which means that they end up playing a role in how signals are sent, depending on how healthy or unhealthy the brain is and things like that. So this is a quick summary figure of the two. For reuptake, what we tend to have is the neurotransmitter. It binds, and then we have an active process that brings it back up into the cell for recycling back into vesicles. On the other hand, enzymatic deactivation, what we have is the neurotransmitter that's released, and then we have some enzyme out at the synapse that takes this and breaks it up into component parts, and then our glial cells and other things will help to get rid of these component parts. And in a way, they're probably going to be recycled again because they're still little pieces of proteins. However, they're not necessarily taken back up into the cell, or at least not in as great of amounts in order to be packaged again. So there's not a recycling process. It's more of a decomposition process. So for me, this last idea is kind of the meat and potatoes. And the reason I say this is because if I were somebody like you that was a wonderful student and you said, I really want to be certain that I understand the action potential to a T, then one of the things you would start to think about is how all of these signals get integrated, right? Because we've now talked about the input at the dendrite, and we now know a little bit about how neurotransmitter is released and what kind of potentials it can cause, whether an inhibitory postsynaptic potential or an excitatory postsynaptic potential. And then you also have learned about the action potential and how that is produced. What happens if the neuron becomes hyperpolarized or depolarized, and how much depolarization do we need to trigger an action potential, and what happens throughout that process. So if you've been paying attention up to this point, you're saying, well, we've got the input and the output. Where's the processing? And that's what we're going to talk about right now. How do we integrate or process synaptic signals? So we have signals coming in, and maybe one neuron could get 5 or 10 or 100 or 1,000 different signals at the same time. And so how do we work these out? Because of course we can have excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And if these come in at the same time, then we should have a very clear outcome, which is that they should cancel each other out. And in some cases they do. The important thing here is how they come together, right? And this is where the job of the soma becomes so important, because if we have a number of excitatory postsynaptic potentials, and usually we need many of them, then what has to happen is that we need them to interact in just the right ways to be able to trigger those voltage-gated sodium channels. We have to be able to hit that threshold of excitation. And in general, it's going to take multiple sources of excitation. In some cases, maybe those kind of local branches at the synaptic terminals onto a few dendrites, or perhaps in other cases, the influence of multiple neurons sending a signal at the same time in order to allow enough excitation from these excitatory postsynaptic potentials to depolarize the soma nearby to that. There are probably two main ways that this happens. One by volume, but the other is something that I hinted at a couple of lectures ago, and I'm pausing here because I'm thinking about how much I want to say about this. But the other is timing, right? So if we've already, this depolarization can happen kind of locally. And if a dendrite's really small, it's going to have fewer ion channels, fewer voltage-gated sodium channels, and so when it depolarizes, we're going to have a little less influence. But if we happen to have that carry a little ways down the dendrite, and then we happen to have another signal come in with the right timing and location, then sometimes together they can have a really great influence. So I think I will leave that idea at that. The one thing I'll say to then bring this all back together is that we think of this process as being kind of arithmetic. And there are a few different pieces that come together to make this arithmetic equation work. So of course, the first is the sum of the positives, or the sum of the positives and the negatives. So if we have just positives come in and there's enough of them to stack up and reach this threshold of excitation, we're going to have an action potential. And of course, as we said, if we have the inhibitory postsynaptic potentials coming in and also excitatory postsynaptic potentials, then you have kind of a negative and a positive, and they're going to come together to cancel out and become zero again. But another thing that is really important here is kind of what I was just hinting at, which is that not only do we have these numbers coming in, but they also have a little bit of a weighting to them. And what I mean by that is that the further away from the cell body, the smaller the dendrite tends to be, and also the more distance that an electrical signal like an excitatory or an inhibitory postsynaptic potential would need to travel. And so if that's the case, then those signals tend to carry less weight. And as we get closer and closer and closer to the cell body, that puts us closer to more voltage-gated channels and all kinds of things like that. So the closer we get to the soma, both the bigger the dendrite tends to be and the more weight it tends to carry in terms of being able to trigger or even shut down an action potential, depending on whether it's an excitatory or an inhibitory signal, respectively. So let's put this all together then. Let's go back to our types of synapse. The axodendritic synapse, which we said is the most common, it could have more weight if it has signals that happen to be closer to the soma, right? If we have on what we would call a proximal dendrite, than if it were on one that was further away. And the other thing is that dendrites, we're going to talk about probably quite a bit later in the semester, but they can there are some processes that change their size and shape that we think happen across learning. And so if we have a dendritic spine that has grown to be bigger, then it can also have more influence. So that's the primary way that these signals are influenced. Now, as you might have imagined from what we just said about something being closer to the soma having more weight, that means that these axosomatic synapses, where we have an axon coming in and making contact on the soma of another cell, these carry incredible weight. And that's probably why there's very few of them. And in fact, they tend to have a very special job. They're more often inhibitory signals that we think of as putting the brakes on some sort of signal that would be coming into the neuron. And we call this either blocking or more often than not shunting an action potential. So if we have a neuron that releases a neurotransmitter like GABA that tends to be inhibitory, it usually opens up chloride channels, and we have that has an incredible influence on the cell body. It keeps it from ever becoming able to reach that threshold of excitation. And when you put all of these pieces together, then you see that having that influence right at the cell body allows it to carry an incredible amount of weight in deciding what the cell is going to do. And that's part of the. So the final thing that we need to talk about are these axoaxonic synapses. And one thing that you have to realize for these is that they don't contribute to this processing step at the soma. Why is that? Because the action potential would have had to have already traveled all the way down this axon to get to the terminal buttons in order for this to be creating some kind of influence. And I suppose there are cases where you could make the argument that then it's influencing kind of the final step in processing or output of the neurons. But What we think these do most often is to do something that we would call either presynaptic facilitation or presynaptic inhibition, which is that they set the tone for what this axon can do. So if I have an inhibitory postsynaptic potential that's coming onto this axon, it's going to make it almost impossible, no matter what the action potential does, for this axon terminal to release neurotransmitter. Or what it might also mean is that when an action potential comes in, I can cause this axon terminal to perhaps release more neurotransmitter, even if I only have a couple of action potentials coming in. So we think of this as being more of an influencer than anything. And then a little surprising piece that I want to bring up just to make you aware that it exists is that there are also dendrodendritic synapses. So if you've been following up to this point, you should realize that this means it's a connection between a dendrite and another dendrite. And there are some very special cases where this occurs, and when an action potential happens in certain cells, the action potential is going to carry all the way out to the axon terminal, but it will also carry through the remainder of the cell. And so some cells, usually ones that are kind of smaller and localized in very special regions of the brain where we have a lot of neurons that need to have crosstalk all the time. For example, sometimes in sensory systems like the olfactory system, then what this allows these dendrites to do is to talk to the neurons next to them and help to modulate the activity of other neurons that are nearby, usually groups of neurons. So this is a synapse that, because it doesn't occur nearly as often as any of the others, we know a whole lot less about, and about all of its different characteristics. So we're not going to focus on it, but I just want to make you aware. I try and break free of the cartoon neuroscience whenever I can and give you every piece. That way, when something like this comes up later on, you're not completely blindsided by it. And finally, something that occurs in very special cases is a type of synapse that integrates signals very differently than most of these others, called a gap junction. So gap junctions, they often occur in the motor system and often in the peripheral parts of the nervous system, but they're electrical synapses where instead of having a true synaptic cleft, they have a direct pore between the two neurons. So you might as well think of this as a bridge between the two neurons. They're perfectly linked to one another. And what this means is that any sort of a change, whether it's a depolarization or a hyperpolarization in the one neuron, is able to have an influence on the other. So you can see an advantage for something like this when we're talking about trying to activate muscle fibers, for example, because then if you send a message out and you want to activate a muscle fiber, then you can activate all of the nearby fibers. And again, just like the dendrodendritic synapses, you're treating these muscle fibers that are close to each other like a group. These kind of synapses occur throughout the nervous system as well, and they're a very direct kind of synapse. And I think the last concept, or very close to the last concept that we need to talk about, is non-synaptic communication. So as I said, synaptic communication is communication that occurs at a synapse, right? Where we have this junction and connection between two neurons, and it's something that's short-lived, and it's basically a direct communication between two cells. On the other hand, we have a lot of neuromodulators and neuropeptides that can signal in different ways. One great example of this are hormones. They're basically a non-synaptic chemical and kind of a broad modulator of what's happening in a cell. And they can be secreted all around the body. Many of them can pass through the blood-brain barrier. And then they're able to come in and affect neurons by flowing through the extracellular fluid, and in this case, they enter into the nucleus if we're talking about hormones or steroid hormones, and they end up interacting with the nucleus to create all kinds of changes in things like protein synthesis and the way that a cell is functioning at some point in time. So they can activate this or inactivate it depending on what the steroid hormone is and where it's working and so on and so forth. So we have these other forms of messages. We're learning a whole lot more about different kinds of RNA that are actually passed from cell to cell like messenger that also have a similar kind of role as this, where they're not necessarily a typical form of synaptic communication, but they influence the cell and communicate between cells nonetheless. So these are, again, a class of signaling molecules that we don't spend very much time talking about. They're kind of cool in what they do, and they're really important to think about, especially for the many of you who said that you want to be PAs and doctors in your introductions. If you're thinking about what's happening when you're giving a steroid, you have to kind of include these sort of things that might be happening in the brain in your thoughts. However, they're also not something that we spend a whole lot of time talking about because it would get complicated very quickly. So here to make you aware of them, and then leave it at that and ask if you have any questions. And per usual, if you have any questions, then there are several different ways that you can reach out to me. You can set up a time to talk during office hours. You can, of course, reach out through Canvas, or you can even use our discussion boards or chat rooms on the Canvas in order to run the question by your fellow classmates. And with that, I hope you have a great rest of the week, and I will see you next time.